There we go. That's better. I'm going to talk to you about a pretty simple idea today, which has really filtered into my thinking over the last few years as we have been developing a new ischemia-activated and highly selective antiarrhythmic drug, and I'll get onto that later on. But one of the big problems in preclinical research, particularly in academia, is the tendency for academics in particular to hype their findings and to move swiftly away from anything negative about something new they're working on. I'm a little bit different from that, which has probably hampered my career. But so I'm going to talk today about the use of the so-called Translational Therapeutic Index in antiarrhythmic drug discovery. So over the years, new drugs, new chemical entities have been championed by discovery scientists, both in academia and in industry. And as I was suggesting a moment ago, clues about safety issues are often downplayed when the efficacy testing is done. So safety pharmacology developed after the tafenidine problem of the 1990s. And very swiftly learnt to try and mitigate the problem of taking forward new drugs that are never going to work by front loading with safety pharmacology studies. But there's always scope for improving efficiency, and for me, efficiency means chucking out the drugs and the pharmacophores that are never going to float because of inbuilt safety issues. So I'd like to suggest that it's possible to facilitate the process of eliminating bad drugs by using the Translational Therapeutic Index. So what is this? It was first mentioned in a paper some years ago. It's not been formally defined, but Muller and Milton said that we use the term to emphasize the need to understand the preliminary translational index as early as possible. And it should be understood that there's a continuum of translational indexes that are refined during the process. And we all understand this. We get a clearer idea about the relationship between the effectiveness and the safety of a drug as we accumulate more data and information. But it may be possible to short circuit this process to a degree by a simple approach of accumulating safety and effectiveness data from the same studies. So the TTI is an animal model variant, if you like, of the therapeutic index. And the upgraded definition I'd like to offer is that it's the ratio between the maximum concentration with no use-limiting adverse drug reactions and the lowest concentration with benefit in a disease model. So data from the same experiment. Now that's the twist, if you like. And I did a Web of Science search, and I only found one paper that had a Translational Therapeutic Index in its title. So this concept hasn't really caught on. It's not captured the attention either of discovery or safety pharmacology scientists, as far as I can see. I mean, maybe it's something that people use and don't publish. I don't know. I've got I can't see my title because the way this thing's set up is there's a whole dialog box at the top. Never mind. So I think it says something like using the Translational Therapeutic Index. So there are some simple ground rules, I think, that could be applied when we use the Translational Therapeutic Index. And the first and the most important is that the safety data that one's generating should be generated in the proof of concept model. So you generate clues about safety and clues about effectiveness in the same experiment with contiguous readout. So this allows threshold concentrations for safety and effectiveness to be compared from a single data set. So, and this, of course, will then obviate the numerous confounds outlined by Muller and Milton, where they're thinking about data from different experiments rather than from single experiments where there are variations in pharmacokinetics and exposure, even worse, species setting and whatnot. So you're lost when you're looking at and this is always a big challenge. You know, I've done a bit of consulting over the years where people show me data sets from numerous different experiments and models, and then say, well, what do you think? Is this a drug, or is it dangerous? I don't know is the answer. So there's an obvious caveat from all of this, of course, and that is that the Translational Therapeutic Index generated from a single model can't be extrapolated. It is self-contained. And of course, what this means is if the Translational Therapeutic Index derived from experiments in a single model look as if the drug has potential to be developed, then one needs to do lots of additional research. But if the Translational Therapeutic Index is found to be narrow or none, this actually provides us with useful information because this can help us make a swift decision about excluding unfeasible drug candidates. So how do we go about using the TTI then? Is there a rubric which we can apply? So the rubric is simple. Never estimate the Translational Therapeutic Index from separate safety and efficacy data. Generate the TTI from a single model. And if the TTI is small or none, that's an important point to consider halting progression. And I've got some data that I'll show you shortly to illustrate this point. Now, if the TTI is large, then clearly it's reasonable then to proceed with further efficacy and safety testing according to the goals and objectives of the company and the program. So can we validate this rubric then using a set of data from a disease-specific database? So let's take a look at this. I can't get my slide to move on. What's going on here? There we go. So here's an example of a data set generating a TTI. So we've been working on antiarrhythmic drug discovery for a very long time, 30 years or more, seeking a drug to prevent ischemia-induced ventricular fibrillation. And as part of this, this process, we've examined countless standard and novel antiarrhythmic agents, generally finding that things are not sufficiently selective in terms of their effectiveness to warrant further work. So as I mentioned earlier on, that it's standard in academia to hype the effectiveness and somewhat downplay the adverse drug reactions in experiments. I've seen this countless times. It even can happen in industry, and I've seen that too. We've tended to take a different approach, and I'd rather be in a position where I can stop wasting time, money, and effort pursuing something that's never going to work. So here's an example case study. So this used our standard bioassay, our kind of entry-level assay, which is an in vitro coronary artery ligation model using the rat isolated Langendorf preparation. There's a review I published on a massive load of data back in the 1990s. So background, we'd previously shown that verapamil was very effective at preventing ventricular fibrillation in conscious rats, but it had loads of adverse effects, AV conduction problems and blood pressure lowering effects. And of course, we know that it failed clinical trials owing to underdosing, which was done to preclude adverse effects. So a typical problem where a drug doesn't have a good TTI. So verapamil, large database, was a good drug to use as a template drug to explore the perfused rat heart model as a TTI generating bioassay. Because obviously, if you're interested in drug hunting, the entry-level model is not going to be a conscious animal model of disease. You want to start out with something simple. We don't like using single cells. The readout is too nebulous. But with the Rat-Langendorf preparation, we have real ventricular fibrillation induced by ischemia as our primary readout. So this was a study then that Andras Farkas did in my lab in the 90s. And he showed that the study was actually focused on a new drug at the time, abefredil. There was quite a lot of excitement about it until it was found to block cytochrome P, one of these cytochromes involved in drug metabolism, and had interactions with every drug under the sun. But the main piece of information from this study focuses on the effects of verapamil. So this is a verapamil concentration response study looking at 5 concentrations from 10 to 600 nanomolar, which is within the bounds of reasonableness in terms of human exposure when the drug was tested. And we can see that verapamil concentration dependently inhibited the occurrence of ischemia-induced ventricular fibrillation, abolishing it at 300 and 600 nanomolar. But it had adverse effects, including, we can see on the right-hand column here, AV block. So I've put a little ring around the 300 nanomolar showing blockade of ventricular fibrillation, but some adverse effects. And we can also see in the same experiments that at an even lower concentration, at 100 nanomolar, we saw increases in coronary flow. So if a drug increases coronary flow in this preparation, it's dilating coronary arteries. And any drug that dilates coronary arteries is going to dilate resistance vessels and lower blood pressure. Every single drug we've looked at has this. If it increases coronary flow, it will do have a blood pressure lowering effect in vivo. So increasing coronary flow is a vasodilator action, and this vasodilator action is going to produce an unacceptable blood pressure lowering. So verapamil had no translational therapeutic index. It had adverse effects at concentrations below those necessary for inhibiting arrhythmias, and that's, it's a powerful lesson. When we published this work, that was part of our focus, that verapamil clearly, if people had done this work 20 years before, we would have seen that there was no chance this drug was ever going to be useful therapeutically. So we generated a similar set of data using looking at sodium channel blocking drugs, or class 1 drugs, mexiletine and lidocaine. So mexiletine was the first drug we formally characterized in terms of its TTI. And we published this a couple of years ago, where we characterized mexiletine's TTI in the rat isolated heart preparation. And we found that the threshold concentration for suppressing ventricular fibrillation was 1 micromolar. But in the same hearts, we saw adverse effects. So this is PR interval. Over time, I won't go into the full details of the complexity of the study. We were changing the potassium concentration, the Krebs, for various reasons at later stages of the experiment. But the PR interval prolongation here with mexiletine is profound and notable. So this is an off-target effect on the AV node. So this means that mexiletine has no translational therapeutic index. So we've conducted further work looking at lidocaine, and the data generated are part of a growing database that we're creating for class 1 antiarrhythmics. And the reason for that is that we're using data to gauge the potential value of a new ischemia-activated prodrug of lidocaine, as I mentioned at the beginning of this presentation, known as OCT 2013. So we published on this last year. So in the Rat-Langendorf prep, we compared OCT 2013, or LNO as it's also known, it's lidocaine N oxide. And we found that the two drugs were equally effective. This is high concentration, 15 micromolar, but this is prevention of ischemia-induced ventricular fibrillation here in the rat isolated perfused heart. But in the same hearts, we noted adverse effects of lidocaine. We see bradycardia on the left in the red with lidocaine, and we see PR prolongation with lidocaine as well. LNO didn't have these adverse effects. So we did conduct, I wish I could see my title. This is going to be a problem for other presenters. They're not going to be able to see their titles because of the dialog box. So in further work we saw then, we did another study with higher concentrations of LNO and lidocaine, much, much higher. And even at these concentrations, there were no effects on PR or heart rate with LNO, and we can clearly see adverse effects of lidocaine. So what are the implications of this? Well, verapamil and mexiletine clearly lack a translational therapeutic index in the Rat-Langendorf prep. From previous studies in our lab, although we didn't present the work in this way, we did report the adverse effects, but we can infer the same conclusions for a range of different drugs, glibenclamide, tadalafil, and all sorts of other putative antiarrhythmic drugs or drugs that affect the pathological process of ischemia. And, you know, the likelihood of anything that people are looking at in preclinical research making it to become a drug, we know it's very low. So the fact that we can't find a translational therapeutic index in our experiments is as one would expect, really. We're not concerned. We think this is actually an indication that the TTI is a very useful piece of readout. So of the drugs that we have looked at over the years, the ones that have been tested for prevention of sudden cardiac death in humans have failed to translate. Generally, it's interesting actually, generally because of lack of effectiveness owing to selection of very low doses in order to avoid adverse effects. So what we're proposing is that the lack of the translational therapeutic index that's been found in studies where effectiveness and safety are evaluated at the same time is a clear indication that the drug will fail. And that, of course, is useful information. So there are caveats, of course. So if a drug's got a promising translational therapeutic index, and I think more than 30 is minimum for most drugs, certainly if we're looking at Torsades de Pointes liability, then this can trigger further discovery research. We have to continue researching on drugs that look promising, of course. And that's because a good translational therapeutic index doesn't prove safety, especially if it's generated using the in vitro model like our isolated heart, because obviously we can't rule out effects on other organs. But this is the good thing. A bad translational therapeutic index can't be made better, even when an in vitro disease model is used to generate the translational therapeutic index. But that, of course, is provided that the disease endpoint is a hard endpoint, not an unvalidated surrogate. So we use ischemia-induced ventricular fibrillation as our surrogate for ischemia-induced ventricular fibrillation. And we are reasonably confident that this is a hard piece of readout. So what of the drug that we've been attempting to develop? We have simple but clear evidence from our isolated heart studies that the TTI is yet to be determined because LNO had no adverse effects up to 60 micromolar. I think the highest concentration we studied in the perfused hearts and 100 micromolar in some other experiments. And we've conducted incremental IV dose studies in anesthetized rats, and the TTI in the perfused hearts compared with lidocaine gave us a strong clue that we may be onto a winner. So we've got patent protection and are exploring a strategy for first in human studies. So I'd like to acknowledge all the countless past colleagues who generated masses of negative findings, drugs with poor translational therapeutic indexes in our standard assay. Sean Rees through to Kat Wilder. Louise Hesketh has done most of the pivotal work with LNO, and she's continuing to work in my lab instead of me. We've been funded by a range of different people, and our key collaborator at the moment is Biotherics and Stefan Ogrzinski. So thanks for your attention. I hope I haven't gone on too long, and I'm happy to answer some questions.
Okay, Thanks a lot, Mike, for this very interesting talk. I must admit, I didn't know anything about the Translational Therapeutic Index before. We have a couple of questions. The first one is, how does the TTI approach translate or fit into the new ICH E14 S7B guidelines draft, if at all?
Well, it doesn't fit into it because it's not being considered, but I think it will be useful for it to be considered. So if anybody has any traction with the people developing the new guidelines, then perhaps it's something they can bring up.
So at what point in the drug discovery process would you think looking at the TTI is a kind of useful parameter?
Well, if you're developing a drug for any condition, the process could begin with high throughput screening to find a ligand, and then you're going to want to test the drug in a disease model. You may want to play around a little bit and look at the pharmacology, but if you can identify an in vitro disease model, because they're much easier to deal with, such as our Langendorf ligation ventricular fibrillation model, then that would be a perfect point to start collecting information that can be used to generate a TTI. So as early as possible is the answer, because essentially when you're looking for effectiveness, you see signals of poor safety, then this is the red flag. And as soon as you start seeing it, this is when you need to start thinking about, do we want to carry on with the pharmacophore, or should we fiddle around with the molecule to find something that doesn't have the adverse effects? So early, as soon as possible, I think.
Yeah. What other in vitro models or techniques other than the Langendorf art do you know of that can be used to sort of predict the TTI?
Well, it depends what therapeutic area you're working in. We're thinking about major organs here. Obviously, isolated brains, not going to be anything we're going to be wanting to work on, although people do use hippocampal slices and whatnot. So essentially, whatever disease area you're working on, if there is an in vitro model that is used in your therapeutic area, then that would be the model to use to generate your TTI. I'm primarily a heart expert, but obviously isolated blood vessels, if you're interested in developing something to dilate blood vessels. I'm not sure there's much call for that these days. We've got enough antihypertensive medicines. Certainly lung, people do use in vitro lung preps. So that's another example, perhaps.
Okay. Well, if there are no other questions, I think we can say thank you very much, Mike, for this very interesting talk.
Pleasure.
And now